So today I'm going to talk about crack channels, and you heard about these. You've already been introduced by Stefan this morning, calcium release activated calcium channels. And I was originally going to talk about astrocytes, but I'm going to change my talk like with many other people. I'm going to talk more about microglia instead, still glia. So just to remind everyone, crack channels terminology in the field is a little bit messy. After the gene was identified by Fesky and colleagues, they called it ORI, and so we know of these channels now. They go by the ORI family of proteins. There are 3 of them in humans. They're highly calcium selective channels, and by some measures, they're thought to be the most calcium selective channels in our bodies. And so when they are activated, you get a very pure calcium current. And the physiological mode of their activation is in response to stimuli that deplete ER calcium stores. Now, physiologically, that can happen in response to a variety of cell surface receptor stimulation, like GPCRs or receptor tyrosine kinases, which generate IP3 that can activate IP3 receptors. Calcium flows out of the IP3 receptor into the cytosol, lowers the ER calcium concentration, and it's that lowering of the ER calcium concentration that then serves as the proximal trigger for activating these channels in the plasma membrane, which is a very fascinating process, which begins with the activation of the calcium sensor in the ER. This is a molecule called STIM1. STIM1 will then migrate to those regions of the ER that are in very close proximity to the plasma membrane and directly interact with the ORI subunits. There is a direct physical interaction between a domain on the cytoplasmic region of STIM1 with the C terminus of ORI1, and that then gates the channel open. Calcium flows into the cell, and that calcium can then drive many different effector functions, and this is a growing list which includes gene expression, migration, proliferation, the immune response, as you heard this morning from Stefan, and so forth. So over the years, I spent, and I would say the bulk of my my independent scientific career, looking at the activation mechanism and the gating mechanisms of these channels. And what we know about the process is actually quite a bit. If one looks back at the entire field, then what we know now is that the STIM1 binding to the ORI-C terminus, which occurs over here in this beautiful structure that was solved by Xiaowei Hu in Steve Long's lab. STIM1 binds over here, and that gating signal is then transmitted, we think, through the transmembrane domains within the lipid layer to open a hydrophobic gate, which is composed of 2 important residues, the valine at this position and the phenylalanine at this position. And the phenylalanine in particular basically stays in a pore-facing orientation in the closed state of the channel and gets displaced away from the ion conduction pathway to lower the free energy barrier for occupancy of the pore with water and ions, and that is the gating process. That's the pore opening mechanism. Now, since those, along with those biophysical studies, we have been really focused, we're increasingly moving towards trying to understand the physiological functions of these channels. And the one organ system where we think that very little is known about these channels is the brain. So that, in many ways kind of represents the last frontier. There are many, we think, potentially similarities between what's going on in the immune system and perhaps some of the immune functions in the brain, but there's certainly more to the story. And so I wanted to just basically talk about that aspect of the work and just highlight, very quickly a couple of things that we have done. And this is by no means, representative of the field. The field has been beginning to look at this question in a much more broader fashion. But a graduate student, Anna Toth, in my lab has been looking at astrocytes, about which we already heard more from Amanda and other people today. And what she noticed is that crack channels are highly expressed in astrocytes. This is a GCAM6 filled astrocyte, and you can see that when she stimulates the cell in the brain slice with a agent that selectively activates calcium signaling in astrocytes but not in neurons, and that is thrombin, which kind of mimics, in many ways as a chemogenetic agent that's selective for astrocytes and not neurons. That stimulates oscillatory calcium signals, which are ORI1 dependent. At least the stimulus evoked response in the frequency of these oscillations is lost when we look at a conditional ORI1 knockout. The implications of that for astrocyte function, looks like I lost my movie over here, but to understand the implications of that, what Anna did was to use synaptofluorin, which is a fluorescent indicator that monitors exocytosis, which has previously been used in neurons, but actually, as it turns out, it works really well in astrocytes as well. So it monitors the fusion of small vesicles, and it's essentially a pH-sensitive GFP that gets quenched. And what you can hopefully see is that when the vesicles get dequenched in the wild-type astrocyte on the footprint, you can see these exocytotic events, which are, I guess, in this monitor, it's kind of hard to see, but these are essentially lost in the ORI1 knockout astrocyte. So one important role of astrocytic ORI1 channels appear to be to regulate gliotransmitter release, which has implications for the regulation of circuits in the hippocampus and beyond. More recently, Mehdi Maneshi in my lab has been looking at these channels also in neurons, where it's also expressed. And in this system, he was specifically interested in the calcium signals that arise, that ORI1 promotes in dendritic spines. And what he found is that when you delete ORI1, either through a whole brain specific Cree or a more excitatory neuron specific Cree driver, then you lose these synaptically evoked calcium signals in the dendritic spine, which are monitored with GCAM7 in this case. The calcium signals were evoked by glutamate uncaging. Compatible with, and alongside with the loss of this dendritic spine calcium signals, what we found is that in these mice, long-term potentiation at the CA3, CA1 synapse is strongly impaired. In fact, it's lost at the 60-minute time point. And going along with that, these neuronal specific ORI knockout mice exhibit a variety of defects in the learning paradigm, but otherwise they're more or less normal. And so things like working memory in the Y maze, spatial memory in the water maze, fear conditioning, these are tasks that are impaired in the ORI1 knockout neuronal from the neuronal knockout side. But what I want to talk about today is a more ongoing project that relates to the role of these channels in microglia. They're also a very interesting glial cell type. They are the primary inflammatory cell, and we already heard about microglia some at this meeting. They play very important roles in numerous disease processes, and microglia have been getting a lot of attention lately in driving inflammation related following brain injury in relation to neurodegenerative diseases, and that's just a very short list. So microglia are very fascinating cells. In the resting state, they have a kind of this ramified morphology with long processes and a small cell body, but following activation in response to a variety of stimuli like stimulation of TLR4 receptors, for example, or following tissue injury, they can adapt in activated or a phagocytic state, depending on what they're doing, wherein the cell bodies swell up, they retract their processes, and there's a lot of interest in trying to understand the checkpoints, the cellular checkpoints that control this dynamic transformation of microglia from a ramified morphology to this activated state, because these microglial shifts basically control nearly all aspects of microglial function, physiological functions. So quite a bit is known about this. This is by no means an emerging topic. There's a lot known, and a lot of molecules have been identified. And among these these include ion channels, which is the topic of this meeting. And the primary ion channels that have received the main focus in microglia are the purinergic receptors, P2X7, P2X4, which is actually not listed on this particular slide, but some calcium channels, metabotropic purinergic receptors like P2Y12 have also received a lot of attention in different physiological and pathophysiological contexts. And the idea is that stimulation of these receptors, as well as other pattern recognition receptors, TLRs and so forth, can drive inflammatory microglial functions by inducing the formation of numerous cytokines and other mediators. It can drive mitochondrial respiration and so forth. Now, when it comes to crack channels, however, ORI channels in microglia, this is an emerging field. Very little is known, and I think The field is still very young. It's underexplored, but a glance at the literature basically shows a few papers. Interestingly, there was one from Reinhold Penner and Andrea Fleick's lab that characterized crack currents that are activated by TLR4 activation in microglial mouse microglial cells. And the idea here is that activation of crack channels might induce the stimulation of calcium-dependent processes, which include things like NF-kappa B and NFAT, but also P38, which is a very important kinase with important roles in microglial function to drive cytokine synthesis and migration. So that was our hypothesis, and we wanted to explore this in the context of a kind of a well-established pathophysiological system where microglia are known to play a very important role, and that is in the induction of chronic pain, and especially neuropathic pain. So I'm not going to introduce neuropathic pain anymore, since you already heard a great deal from Amanda and many other people, Rajesh and so forth. But suffice it to say that microglia have very well-established important roles in the etiology of neuropathic pain. And so our hypothesis is that if we conditionally delete ORI1 in microglia, then it might impair the production of some of these inflammatory cytokines, which can drive the underlying pathology in terms of the remodeling that occurs in the spinal cord and beyond. So we made a microglial-specific ORI1 knockout mice by crossing CX3CR1 Cree with ORI-flox mice. And verified that this is a knockout. We're looking at the mRNA and so forth, both in cultured microglia as well as in freshly isolated microglia from the spinal cord. And ORI1 is lost, and there is no change in the expression of the other ORI or STIM isoforms. And we then started looking at calcium signaling induced by this well-known activator of microglia, which is ATP. So application of ATP evokes a store-operated calcium entry, and the way we study that, and I think Stefan might have already walked you through this, is to deplete stores first with tapsigargin to release calcium from the ER, and then crack channels get activated. So when you restore extracellular calcium, you get a big surge in the cytoplasmic calcium concentration, and we monitor the rate of influx over here to quantify the SOC amplitude. And you can see that blockade of crack channels with certain well-known pharmacological blockers, inhibitors, basically strongly impairs it, but so does conditional deletion of ORI1 in microglia, in this case in a tamoxifen-inducible fashion. This provides kind of like a physiological stimulus where we can then begin to relate this to neuropathic pain and some of the underlying pathology. It is also inhibited by, let's see, I want to highlight CM4620, which is that compound that Stefan talked about earlier this morning. This is a compound that's currently in clinical trials for mitigating inflammation in pancreatitis as well as COVID. I should also point out that there is no real difference in the amplitude or the other properties of calcium signals between male and female microglia, and we thought this was important to look at given the very sex-specific contributions of microglia to neuropathic pain. So deletion of ORI1 basically blocks store-operated calcium entry in both cases. I think the previous slide was showing tapsigargin. But so here is ATP, SOC evoked by ATP. You can see that if you either use ATP or a more P2Y specific ligand like UDP or ADP, you also get store-operated calcium entry, which is lost in the ORI1 knockout. And so we then began to wonder, what is this good for? And basically took a page out of many of the types of assays that Stefan does in his lab, and we looked at cytokine induction, and we looked at a variety of pro-inflammatory cytokines that are implicated in the etiology of neuropathic pain, including IL-6, TNF-alpha. Prostaglandin E2 is not a cytokine, but it's produced in a de novo fashion by activation of CPLA2 in many different cells. And in each case, you get strong induction either by activation of if a ligand that stimulates the production of these cytokines, or by direct activation of crack channels with tapsigargin. And this induction it's not completely lost, but it's greatly diminished in the ORI1 knockout cells. And this is important because, as the field has previously shown, direct application of cytokines like TNF alpha, for example, uh, is known to increase the synaptic transmission in the dorsal spinal cord of excitatory neurotransmission. And so here is our rendition of that type of experiment, which is very well documented in the literature, that application of TNF alpha increases the frequency of spontaneous excitatory postsynaptic currents when you're recording from substantial gelatinous neurons in the dorsal spinal cord. So what happens when we actually do a neuropathic pain model? Well, so that's summarized over here. I'm not going to introduce neuropathic pain since I think we already heard quite a bit about this, but we use the spared nerve injury model where we ligate or transect a couple of branches of the sciatic nerve while leaving one of the branches intact. And so that is a very robust model of neuropathic pain where the mouse has increased sensitivity to mechanical stimuli that would otherwise be not perceived as pain. The mouse now perceives it as pain. And so when you look at von Frey thresholds, paw withdrawal thresholds, you get a sharp decrease in the threshold where the paw is withdrawn, and we are just quantifying the force on the y-axis for that paw withdrawal. And what we see is that compared to the sham-operated controls, the ORI1 knockout mice show some mitigation of this neuropathic pain. It's highly significant compared to the wild-type mice, but it's certainly partial. So this is not by no means a complete protection. And so that told us that ORI is, in fact, in the pathway that mediates neuropathic pain. But surprisingly, we saw this mitigation only in males. In the females, there was absolutely no effect. So you can delete ORI1, and there's no protection at all whatsoever. So this is, these kinds of things have been described for microglia in the past, but most of those experiments were dealing with poisoning microglia or inhibiting them with minocycline and so forth. So we felt that this is a little more of a precise genetic perturbation where it kind of recapitulates that sexual dimorphism. And I think it also kind of points out that this is more, not related to neurons, but more a microglial phenotype because the perturbation is only in microglia. We wanted to understand this a little bit further. And so Shogo Tsujikawa, who's a postdoc that did this work, began to look at cytokine levels in the bulk spinal cord in the homogenate. And there was a significant reduction of several of these cytokines that have been implicated in neuropathic pain, and which we noticed were produced in an ORI1 dependent manner in isolated cultured primary microglia, which included things like IL-6. You can see here that the enhancement of IL-6 following nerve injury is basically completely abolished, and the induction of some of the other mediators was cut by more than half. Again, this effect was completely specific to males. There was no effect whatsoever in the females. And so that was, again, a head-scratching moment. He then went in and did electrophysiology in spinal cord slices, recording from substantially gelatinous neurons. And there is this well-described effect where the frequency of excitatory synaptic transmission increases following nerve injury. And so that is what Shogo also noticed, is that there's a significant increase in the frequency. And this frequency enhancement was blunted in the males, again, only, but not in the females, as you can see down here. So at every level, starting from the bulk cytokine levels, synaptic transmission on the spinal cord, there appear to be this sexual dimorphism, but it doesn't appear so when you take the microglia out of the animal and you're looking at the induction of cytokines in cultured cells. So I'm not here to tell you the answer for what's the basis of this sexual dimorphism. That's something that we would like to understand further, but we think it might have something to do with sex hormones. And I can tell you that when you do this experiment in, let's say, gonadotropized mice, then the sex difference disappears. Well, what's going on? In terms of a little more detail, when we looked at microglial proliferation and activation, one way that you can actually assess this in the intact spinal cord is by IBA1 labeling, which is a ionized calcium-dependent protein that's expressed selectively in microglia. And what you can see is that in the sham-operated state cells, in the slices, the microglia kind of look much more reminiscent of those ramified resting state microglia with long processes and smaller cell bodies. And there's a pretty obvious um, difference in the following SNI, where they adapt a much more swollen cell bodies. Not only that, the area significantly increases. We think that when you quantify the area as well as the number of microglia based on the DAPI1 stain, both of them significantly drop, but again, only in the males, but not in the females. So microglial activation appears to be ORI1 dependent, at least partially in the males, but it doesn't appear to be ORI1 dependent in the females. This is just a slide showing that the expression of ORI is not changing in response to SNI. And I'll just finish by saying that we also looked at this CM4620 compound to see whether a small molecule inhibitor that's currently in clinical trials would offer any mitigation of neuropathic pain. Let me just jump ahead here. And what I want to show you is that as long as we apply, when we dose the mice, and this is delivered by oral gavage, there is some protection in the males, and it's actually really obvious when you induce neuropathic pain first, and then you look at the mitigation of neuropathic pain or mechanical thresholds after the fact. But the protection lasts only for as long as the drug is delivered, and it completely reverses soon after that. Effect is completely unique to only males again, doesn't offer any protection whatsoever in the females. So at every step along the way, it seems to be the case. If we give the drug to microglial conditional ORI1 knockout mice, in male mice, there is no protection. So the protection depends on the presence of the drug target in microglia. And if you delete the drug target in microglia, then even a systemic injection of the drug basically has no effect. So these are the conclusions. I'm not going to go through the list, but basically just acknowledge the people that did this work. This project was primarily driven by Shogo Tsujikawa, who is a postdoc in my lab, former postdoc, and Kate, who's a current graduate student. And we collaborated with Mike Salter in Toronto for most of this work, because he's really an expert in neuropathic pain. Thanks.
Very nice. Great job on timing. We have time for one or two questions. If there's no question yet. Okay.
So the CM4260 is not specific to only blocking in microglia, right?
Exactly right. So that's what was interesting, is that the drug goes everywhere, presumably, and so we wondered, why do we get what appears qualitatively to be a microglial type effect? And it appears to be microglial effect because when we delete ORI in microglia, then the drug target is gone only in microglia. So you may be wondering, why is this the case? Given that ORI, I just told you that it's also in astrocytes, it's in neurons, it's certainly in lymphocytes and so forth. I don't know the answer to that. It might have something to do with the target occupancy and the target occupancy necessary to give you a particular physiological effect. So it may be that the level of inhibition you need, let's say in neurons, to get a non-specific effect, if you will, may be higher. Perhaps you need more occupancy, whereas that in the microglia, and this is very common in the pharmaceutical world, is that you can get 90% blockade in one particular cell type and you'll get a phenotype, but in a different cell type, 10% blockade may be enough. And so I think that's what it may be. That's what I'm guessing.
I'll ask you mine afterwards. I think I know why I got this job now, is to move them on. So, okay, in the interest of time, thank you, Murali. That was excellent.